in human CD8 T cells. Thank you, Firas.
Thank you for the introduction. Good afternoon. So our lab's focused on understanding the expression regulation of the interleukin-7 receptor and interleukin-7 or IL-7 signaling. So to talk briefly about the importance of interleukin-7 signaling in the, for T cells. So IL-7 plays essential, non-redundant roles for the development, for the survival, as well as for the function of T cells. And that's illustrated by a diagram. So it promotes proliferation of these T cells by upregulating genes such as BCL2 and downregulating apoptotic genes such as BAX and BAD. It also is heavily involved in the antigen-specific CTR response of T cells, especially and clonal expansion upon encountering a foreign antigen, such as an HIV protein, as well as mediates the CTR response, so in the form of perforin as well as granzyme B release, and plays very important roles for establishment of memory. However, for IL-7, in order for IL-7 to signal, it has to bind to its receptor, and the IL-7 receptor is comprised of two units, the IL-2 gamma chain as well as the IL-7 receptor alpha chain, to which it specifically binds to. Interestingly, the expression of IL-7 receptor alpha chain, which I'm going to refer to as CD127 from now on, it is severely dysregulated in HIV infection. So, in layman's terms, the HIV infection downregulates the expression of this important receptor and renders these T cells very ineffective to fighting the infection. So, I've listed a few facts here. Levels of IL-7 are much higher in HIV-positive individuals, as well as currently IL-7 has been examined in several clinical trials to correct lymphopenia in BMT as well as in chemotherapy and HIV infections. So the targets of our studies are trying to understand how IL-7 downregulates the expression of this receptor, and after that we'll see how HIV intersects with the process. What we know so far is that interleukin-7 downregulates the expression of CD127 at the transcriptional level, and I presented that on Monday in detail. And here I'm presenting the story at the protein level. So IL-7 downregulates the protein expression as measured by Western blot analysis in a time-dependent manner. And this downregulation is dependent on the proteasome, as illustrated by utilizing the MG132, the 26S proteasome inhibitor, which blocks the IL-7 mediated downregulation of CD127 protein. So the big question is, what mediates this process and what targets this protein to the proteasome for degradation? So long story short, we came across one of the families of proteins that are involved in mediating the expression of several cytokine receptors. So they are the SOX proteins, the suppressor cytokine signaling proteins. There are 8 members, so it's a family of 8 members, SOX 1 to 7 and cis. And they are known to recruit ubiquitin ligases. So they bind to proteins, recruit ubiquitin ligases, and the complex is targeted to proteasome for degradation. So it was an interesting and likely candidate to examine. So the first question we asked, are these expressed at all in human CD8 T cells? So we measured the basal levels and we saw that they are expressed, and the expression is at varying levels. So the relevant question now is, does interleukin-7 upregulates the expression of any of these SOX transcripts? In fact, interleukin-7 induces the expression of 3 of the 8 members of the SOX family of proteins. At the transcriptional level, cis, SOX1, and SOX2, and the induction occurred in a time and dose-dependent manner, with a maximal induction level at 3 hours, and that's pretty much the time which we see the downregulation of total protein of CD127 at the protein level to occur. So the next question, before we find out the relevance of of the inductions and CD127 downregulation, is how does this induction occur at the mechanistic level? So we utilized a number of inhibitors of the signaling pathways that are known to be induced by IL-7, namely the PI3 kinase, the JAK, and the STAT5 signaling pathways. By doing so, I was able to show that the induction, the IL-7 mediated induction of these three genes transcripts is JAK dependent, because by inhibiting JAK signaling, I was able to block the induction of these transcripts. Similarly, I showed that it is STAT5 dependent, and this signaling pathway is known, the JAK-STAT signaling pathway is known to activate the transcription of genes. So we wondered if the induction of the SOX transcripts was direct or indirect, did it require a new protein synthesis or not? So we pre-treated CD8 T cells with cyclohexamide, a translational inhibitor, and that did not block the induction of SOX mRNA, suggesting it is direct. Collective, as well as we mirrored the induction of the transcripts at the protein level. So collectively, this is the mini model we have. Interleukin-7 binds to its receptor, activates the JAK STAT5 signaling. STAT5 signaling is known to induce the phosphorylation of STAT5, which translocate the nucleus and activate the transcription of these three SOX genes, which have STAT5 binding sites and their promoters. So next question, well, do these induced proteins interact with CD127? So we examined that by co-immunoprecipitation, and saw that these three proteins physically interact with CD127 protein. Only cis and SOX2, however, the physical binding increases in response to IL-7 signaling. So the next question is to confirm these findings, as well as to elucidate the localization in CD8 T cells at which this binding occurs. And we did so by confocal microscopy. So when we, in the panel A, we measured the co-localization of CD127 with cis, SOX1 and 2, and obtained a very similar conclusion to the co-IP data. However, interestingly, we found out that the interaction of cis SOX2 with CD127 occurred in early endosomes, as measured in the EEA1, the early endosomal marker. So it does not occur at the receptor level, but occurs downstream with that. And when we measured if the targeting of the SOX proteins is shifted towards lysosomal degradation or perosomal degradation, we looked at markers of the lysosome, so LAMP1, as well as the proteasome, MCP20. And what we saw is that the co-localization, there is not much co-localization with LAMP1, but with MCP20, further confirming that these SOX proteins, once are induced and bound to CD127, are targeted to the proteasome for degradation. And this was further confirmed by utilizing a specific inhibitor to ubiquitin ligases, which showed that, which blocked the IL-7 mediated degradation of CD127, as well as resulted in the accumulation of cis and SOX2 proteins. And lastly, the last piece of data I want to show as well. So lots of data here, but the conclusion is we also measured the contribution of these three proteins in the IL-7 mediated regulation of CD127 by siRNA mediated knockdown assay, and showed that there seems to be a functional redundancy between SOX2 and cis in the downregulation of CD127. Collectively, this is the model we are putting forward. IL-7 binds to CD127, which induces the rapid internalization and phosphorylation, which we showed in another study, as well as it initiates the JAK-STAT5 signaling pathway, which induces the expression of cis and SOX2 mRNAs and proteins, which interact with the internalized CD127 receptor protein in early endosomes, and then the complex is targeted to the proteasome for degradation. At the end, I would like to acknowledge my lab colleagues as well as the blood donors and the funding agencies. Thank you very much for your time.